ꯑꯩꯈꯣꯏ ꯉꯁꯤꯒꯤ ꯇꯣꯄꯤꯛꯁꯤꯗꯤ RNA ꯋꯥꯜ ꯍꯧꯗꯣꯏꯅꯤꯀꯣ DNA ꯂꯣꯏꯈ꯭ꯔꯦ ꯍꯧꯖꯤꯛ RNA ꯁꯦ ꯍꯧꯗꯣꯏꯅꯦꯕꯀꯣ ꯑꯗꯨꯅ ꯑꯩꯈꯣꯏꯅ ꯃꯃꯥꯡꯒꯤ ꯗꯤꯁꯀꯁꯟ ꯇꯧꯈ꯭ꯔꯤꯕ ꯗꯤꯁꯀꯁꯟꯗꯨꯒꯤ ꯃꯇꯨꯡ ꯏꯟꯅꯀꯣ ꯐꯥꯔꯁ ꯖꯦꯅꯦꯇꯤꯛ ꯃꯦꯇꯔꯤꯌꯦꯜꯁꯦ ꯀꯔꯝꯕꯅꯣ ꯍꯥꯏꯕ ꯀꯣꯏꯁꯟꯁꯦ ꯂꯥꯛꯑꯦꯕꯀꯣ ꯐꯥꯔꯁ ꯖꯦꯅꯦꯇꯤꯛ ꯃꯦꯇꯔꯤꯌꯦꯜꯁꯦ ꯀꯔꯝꯕꯅꯣ ꯍꯥꯏꯕ ꯀꯣꯏꯁꯟ ꯂꯥꯛꯑꯦꯕ ꯑꯗꯨꯅ ꯁꯤꯒꯤ ꯀꯣꯏꯁꯟꯁꯤꯒꯤ ꯃꯇꯨꯡ ꯏꯟꯅ ꯗꯤꯇꯦꯜ ꯗꯤꯁꯀꯁꯟꯗ ꯑꯩꯈꯣꯏꯅ ꯁꯤꯒꯤ ꯇꯣꯄꯤꯛꯁꯤꯗ ꯇꯧꯗꯣꯏꯅꯦꯕꯀꯣ ꯑꯗꯨꯗꯤ ꯌꯦꯡꯁꯦ RNA was the first genetic material. Genetic material ꯈ꯭ꯋꯥꯏꯏꯍꯥꯟ ꯍꯥꯟꯕꯁꯦ RNA ꯅ ꯑꯣꯏꯌꯦꯕꯀꯣ. There is now enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc. ꯑꯗꯨꯅ suggest ꯇꯧꯕ ꯃꯇꯝꯗ ꯑꯩꯈꯣꯏꯒꯤ life processes ꯍꯥꯏꯗꯤ ꯑꯩꯈꯣꯏꯒꯤ lifeꯁꯦ ꯒꯤ ꯃꯔꯨ ꯑꯣꯏꯕ ꯄ꯭ꯔꯣꯁꯦꯁꯁꯤꯡꯁꯦ ꯑꯗꯨꯒꯤ evidence ꯑꯣꯏꯅ ꯍꯥꯏꯕꯗꯤ metabolism, translation, splicing ꯁꯤꯅ ꯁꯤꯒꯨꯝꯕ ꯃꯌꯥꯝꯁꯤꯒꯤ ꯁꯤꯁꯤꯡꯁꯦ ꯄ꯭ꯔꯣꯁꯦꯁ ꯉꯥꯛꯇꯅꯤꯀꯣ ꯑꯩꯈꯣꯏꯒꯤ ꯂꯥꯏꯐꯁꯤꯒꯤꯀꯣ ꯌꯥꯝ ꯆꯪꯗ꯭ꯔꯕ ꯌꯥꯗ꯭ꯔꯕ ꯃꯔꯨ ꯑꯣꯏꯕ ꯄ꯭ꯔꯣꯁꯦꯁ ꯉꯥꯛꯇꯅꯦꯕꯀꯣ ꯑꯗꯨꯗꯣ ꯗꯤꯁꯀꯁ ꯇꯧꯕꯗ evidence ꯄꯤꯕꯗꯅꯦꯀꯣ ꯁꯤ ꯈꯪꯕꯁꯤꯅ ꯍꯥꯏꯕꯗꯤ RNA ꯁꯤꯅ first genetic materialꯅꯤ ꯍꯥꯏꯕꯁꯦ ꯈꯪꯕꯁꯤꯅ ꯁꯤꯅ evidence ꯑꯣꯏꯔꯛꯑꯦꯕꯀꯣ evolve around RNA ꯁꯤꯒꯤ ꯃꯌꯥꯝꯁꯦ ꯑꯩꯈꯣꯏꯒꯤ life processes ꯃꯔꯨ ꯑꯣꯏꯕ life processes ꯁꯤꯒꯤ ꯍꯥꯏꯕꯗꯤ metabolism, translation, splicing ꯁꯤꯒꯤꯁꯦ ꯁꯤ ꯃꯌꯥꯝꯁꯦ RNA dagi evolve ꯇꯧꯔꯛꯄ ꯉꯥꯛꯇꯅꯦꯕ RNA dagi ꯂꯥꯛꯈꯤꯕ ꯉꯥꯛꯇꯅꯦꯕꯀꯣ RNA used to act as genetic material as well as catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalyst and not by protein enzyme ko RNA se genetic material oina act tawre haibadi ma se first genetic material ni hare aduna RNA se genetic material oina act tawrabani na ma se catalyst oina act tawi hadi catalyst haibadi kaino haba matamda biochemical reaction ama thokpa matamda madubu boost tawriba ko hadi activate tawriba do RNA na tawhaleba ko hadi akoigi living system sida ꯑꯩꯈꯣꯏꯒꯤ living system sida RNA ama ꯍꯥꯏꯕꯗꯤ reaction biochemical reaction ama ꯊꯣꯛꯄ ꯃꯇꯝꯗ ꯑꯩꯈꯣꯏ ꯏꯁꯥ ꯃꯅꯨꯡꯁꯤꯗ reaction ꯃꯌꯥꯝ ꯊꯣꯛꯏꯅꯤꯗꯅ ꯑꯗꯨꯒꯤ reaction ꯗꯨꯒꯤ ꯃꯇꯨꯡ ꯊꯣꯛꯍꯜꯂꯤꯕ reaction ꯗꯣ ꯀꯅꯥꯅ boost ꯇꯧꯒꯦ ꯍꯥꯏꯗꯤ RNA ꯁꯤꯅ boost ꯇꯧꯋꯦꯕꯀꯣ ꯍꯥꯏꯗꯤ material ama genetic material ama oina mase catalyst oina su act tawi habanida ko mana sida haise adubu protein enzymes na di nateba adudo boost tawriba catalyst oise ko catalyze tawrise but RNA being a catalyst was reactive and hence unstable haibadi ko akoina hozik ngasai haigibado ko RNA se catalyst oija tarsu ko ma se reactive yam thokman bana ko hadi reactive oiman bana ko reaction se yam thuna boost tawman bana ko maram oiragana ma se unstable su oiba hadi stable oide DNA ga RNA ga yengna tardi kanana stable oiba henge haiba matamdadi DNA na heleba ko akoi mamangi video da haikrabani ko ani sigi difference so RNA ga DNA gi difference to ko therefore DNA has evolved from RNA with chemical modification that make it more stable. Haibadi RNA se unstable oiba ko RNA se unstable oiba maramna DNA se RNA de evolve tawrakpani haibadi RNA dagi tamna evolve tawrakpani ba adugi matung ina stable oirakpani ba ko DNA se su RNA de evolve tawraga stable oirine ba ko kai kamaina evolve tawrake hai tardi chemical reaction myam thoidana adudo modify tawra adugi chemical reaction do modification tho matungda RNA dagi DNA evolve tawrakpani ba ko aduna stable oibasu helhalakpani ba DNA sida DNA being double stranded and having complementary strand further resists changes by evolving a process of repair DNA se double stranded ne hair se akhoi khange ko adubu RNA di single stranded ne ba ko RNA di single stranded neba ko DNA na double stranded neba aduna having complementary strand magi complementary strand laidana ko haibadi adenine ga thymine ga se complementary natra asi se complementary strand har khangna ba natra khoina adudo resist further resist changes of evolving a process of repair haibadi sigi strand double stranded hec form tawkhiba matam dadi ko moigi evolve tawpagi process to change lakpai process se resist taweba thingani ba ko double DNA se hek stable oikhra matung dadi ko complementary strand sing duna ne change tawpagi chance to process to thingani ba ko next topic replication aina mamang da so hai ke replication haibase kai neba copying haibani ba copy tawba ko copying replication haibadi copying haibani ba ko adudi horasa ko replication while proposing the double helical structure of DNA, Watson and Crick had immediately proposed a scheme for replication of DNA while proposing. That means DNA ki double helical structure propose sharing agi manung dane Watson ga Crick ka sinane khudakta haibadi adum ikan kanadana ne mana DNA gi replication sisu propose scheme se kamaina replicate tauge haibasisu propose tawkhiyeba ko haidi DNA copying se kamaina tawkhige magi duplication se kamaina tawi haibado mana ka hatare immediately propose taweba ko to quote their original statement that is as follow moigi statements the pire ko moi statements se Watson ga Crick gi propose kiva scheme dugi statements se the pire sineba ko it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick, 1953. Moina haise kaino haiba matamda moigi mindset se haidi moina double helical structure of DNA se propose storing ai da moi mindset se escape tawdeba haidi adugi mahutne moina khudakta immediately postulate kai postulate tawe DNA dugi that means DNA habadi genetic material habana tro genetic material dugi copying replication thokpagi mechanism moina ikan kanadana postulate tawbanga mi habani ba ko moigi statement se moina piriba statement se the scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strand. Scheme dugi moina suggest about that means Watson ga Crick kana suggest riba scheme da aduda moina suggest risa kaino hai tari DNA ki strand ani laidana adugi strand do separate tawbirani ba ko tongan separate tawbirani sigi strand ani se strand ni separate tawbira ko si singsina parent oina act tawrani ba template haidi parent oina act tawbudune ko template sisu parent amo oina act tawrani ba sina parent oraga anauba amuk complementary strand amuk moina produce tawdone ba ko new strand amuk produce tawdone ba ko sinasu new strand amuk produce tawdone ba haidi synthesize tawdone ba ko after the completion of replication that means copying haisi duplication se loiraba matung da ko loiraba matung da its DNA molecule would have one parental and one newly synthesized strand haibadi replication the loiraba matung that means copying the loiraba matung da ne ko DNA molecule khuding makine parental haibadi parental strand namaga anauba newly synthesized strand dumaga yauni bako DNA molecule khudingi laini bako adugi scheme dubu semi conservative DNA replication kaweba haibadi semi conservative haidi kaino haiba matamda magi conserve taw my parent amasu conserve taw adum yaudra parent to demand de na magi new strand am puthokpa matamda magi daughter strand am puthokpa matamda magi parent si demand de na parent to su yauna magi new strand to su yaure ena ne semi conservative DNA replication haina kaurambane ba ko moina kaurami se magi experimental proof amuk yeng se ba ko experiment tawraga mana proof su tawrame ba ko it is now proven that DNA replicates semi-conservatively. It is now akoina proof tawkhre ba ko DNA se semi-conservatively replicate tau hai di copy tau ba ko semi conservatively that means parent strand amaga magi daughter strand amaga yawe habane ba ko ani mag pull up yaw min nei habane ba aduna semi conservative ko ba natra it was shown first in E. coli and subsequently in higher organisms such as plants and human cells. Matthew Meselson-Stahl and Franklin-Stahl performed the following experiment in 1958. Ngasai scheme suggest tawkibduna Watson ga Crickna 1953 da suggest tawbako. Aduga huzik adugi proof oina experiment tawbasina Matthew Meselson-Stahl ga Franklin-Stahl gana perform tawbani ba adu sige experiments edusu 1958 da ko. First point they grew E. coli in a medium containing NH4Cl ammonium chloride haibadi E. coli da nego experiment se E. coli bacteria sidane bacteria si NH4Cl yawba medium amada grow tawhalle ko and N15 is the heavy isotope of nitrogen. Nitrogen ke isotope like 40 N14 haibama N15 haibama aduda sida use story se N15 sineba ko N15 se heavy isotope har khangnei bo nitrogen ki damakta ko as the only nitrogen source for many generation haidi generation mayam gi damakta nitrogen source ma khataneba lairisi N15 se ko the result was that N15 was incorporated into the newly synthesized DNA huzik growth of hankre ko E. coli se N15 that means ammonium chloride yawba medium of the growth of haleko aduga my result se kai phangake haiba matam the N15 se now na synthesize tarakabda now na puthore iba DNA se sida yaureba N15 se as well as other nitrogen containing compounds as well as other haidi nitrogen yauba ate topa compound me amasu yauba adugi haidi anau na synthesized puthora iba DNA sida ko this heavy DNA heavy nitrogen yaurabanina mas heavy DNA oifam thoktra aduna this heavy DNA molecule could be distinguished from the normal DNA normal chumnagi ne normal DNA se normal DNA sidagi ne normal DNA sidagi heavy heavy DNA haidi N15 yauba ko N15 yauba DNA se sise haidi khaidok payai kamaina centrifugation in a cesium chloride sida ne centrifuge tauragande normal DNA dagi heavy DNA se khandok payai ba distinguish tobe to khai thambiyai ba ko density gradient please note that N15 is not a radioactive isotope and it can be separated from N14 only based on density. Haibadi note neko important neko asise haibadi N15 se mase radioactive isotope nate ba madi heavy isotope haiba khatane ba ko radioactive oiba nate ba aduna N14 se N14 haidi N14 isotope tagina separate tawba yai bako kam ilai na haibadi separate tawba yai kari da base tawraga magi density da base tawraga ko point number 2 adugi matung then they transfer the cells into the medium with normal and 14 that means NH4Cl and took sample at various definite time interval as the cell multiplied and extracted the DNA that remained as double-stranded helices. That means ngasagi adugi matungda na cell haibadi ngasagi N15 DNA se heavy DNA se kaida transfer tawre karamba medium da muk transfer tawre normal NH4Cl da na transfer tawre ko aduga sample lawre karamba sample definite time interval haidi matam akak na ba tongan ba different time interval da ne moina sample lawe ba ko kai tholaki yenge ba ko aduda DNA extract tawrok podo karamba DNA extract tawrok ke yeng doine ba ko aduga double stranded helices me amama haidi extract tauraktarsu DNA tongan ba DNA heavy oigera light oigera hybrid oigera haidi DNA se extract tauraktarsu ne DNA do adum double stranded oini ba haidi hong oiba ko stable adum oira sakani ba double stranded oira sakani ba. The various samples were separated independently on cesium chloride grazing to measure the densities of DNA. The various samples were separated independently. Separated, that means ngasagi the different time interval the ne low riba samples sing do independently measure to eba density DNA ki density the base tawraga haidi cesium chloride gradient da ko yeng se bako magi sigi se magi diagram se yeng se bako ahanbada ma se heavy and DNA lore ko heavy nitrogen DNA lore DNA loba matamda time interval da ni ko ahanbagi time interval se 20 minute yeng 20 minute yeng ba matamda hybrid form tore to hybrid ne ko la ise haibadi N14 su N14 generation ahanbagi generation 1 sida N14 su phange aduga N15 DNA su phange aduna hybrid hai ni ko sida hai se hybrid hai ne sida na heavy ne ba ko si se heavy ne ba ahanbagi heavy DNA se lauba matamda 20 minutes se yengba matamda hybrid phange adudagi amuka 40 minutes amu yenge 40 minutes yengba generation 2 generation 2 lakpasda normal DNA su phange light DNA ko hybrid su phange hybrid ani phange aduga light DNA ani phange ko sida phangi se light DNA ani phange ko sida light amaga light ani phange ko aduga hybrid ani phange sine ba haidi different time intervals da moina experiment tawba matamda DNA se extract tawrakhiba se khenarabadi ai heavy phangkhibi ai hybrid phangbi ai light phangbi ai adubu mai se double stranded ti hongde bas double stranded ti adum oiraga chatleba ko mai helical structure do ko aduna ma se stable oita ineba ko magi double stranded adum oiraga chatli do aduna magi figures separation of DNA by centrifugation haidi DNA se separate tawrini ko centrifuge tawraga sigi process se sigi experiment se. Point number 3, thus the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium, that is after 20 minutes, E. coli divides in 20 minutes. Haibadi huze aina explain tawkhri dune ko manasa DNA se heavy DNA loba matamda heavy DNA dagi light DNA da haidi transfer tawba matamda E. coli haidi 20 minute ko 20 minute yengba matamda neba ko had a hybrid or intermediate density haibadi 20 minute ki matungda yengba matamda hybrid DNA phangi haibadi hybrid phangi haiba matamdi 15 heavy DNA maga duga light DNA si ani se pulaga natro hybrid semi se adune hybrid phangi haine bako aduga DNA extracted from the culture after the another generation, that means atopa generation, that means second generation do after 40 minutes yengba matamda generation second generation do ko was composed of equal amounts of this hybrid DNA and light DNA, that means light DNA ga ko equal amounts of hybrid DNA phange ba ko haibadi hybrid ani aduga light DNA ani phange haibane ba ko after 40 minutes se yengba matamda. Very similar experiment involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed on Bisha Faba beans by Taylor and colleagues in 1915. So maina chap manaba experiment se ko Bisha Faba beans na tawramme ba ko kanana Taylor ga magi colleagues sing na ko 1958 ta tawramme ba adusu radioactive thymidine lauraga ko DNA से chromosome da of newly synthesized DNA से chromosome da nauna DNA synthesize तौरक् से radioactive thymidine ना हपचलगा ने experiment से तौरमे बा को सिददी detail याउद्र को लारिक तदी को सिगी level हाइदी class 12 के ओइना दी सिसे detail दे याउदे को थोइना सु important ओइदे को the experiment proved that the DNA in chromosome also replicates semi-conservatively मागी experiment सिदसने chromosome da yauba DNA se copy tawbase semi conservatively tawi habane ba aina hanjan hai se semi conservatively haibadi parent DNA ama su yauraga aduga nauna synthesize tawrpa daughter DNA su yawe habane ba ko daughter DNA sa ani pulaga kaise me habane ba ko semi conservatively DNA same habane ba ko parent DNA su aduga newly synthesized DNA su sine ngasigi topic se ko RNA wall aduga magi experimental proof ko aduna mathangi videos de khoina magi machinery ga enzyme ga tawdoni aduna video se gyan ta daba doubt label resu comment section da dum hairo natraga against da DM dum tawro ko insta gi link su aina description da hapake aduna aigi video se pambi pe yaragadi like share subscribe tawba ko bigano thank you